Hello everyone and welcome to our NGS-CN Summer School online collection on spatial transcriptomics. Before we jump to our next speaker, let's take a look once more who we are. So it's a next generation sequencing competence network that consists of 4 production sites as you see on the map. So in 2019, DFG has funded centralized sequencing applications. There were quite a lot of calls, and within each call there were a lot of projects. And from 2024, DFG has changed the strategy towards individual sequencing applications. That means that now there is no special deadline for a centralized call, and you can co-apply at the DFG together with the chosen genomic facility. If you wish, here is a QR code, contact us, and then we could take a look and maybe sequence together. If you want to know something more about our news and upcoming events, then you also can take a look at the QR code, scan it, and get into our mailing list. Let's grow community together. And with that to be said, let's welcome our next speaker.
Thanks a lot for the opportunity to giving a talk here. So I made these talks a bit more as an introduction. So I'm working in the core facility of genomics in Tübingen, which is like the core facility doing genomic studies at the University of Tübingen. We are also part of the NGS Competence Center in Tübingen. I'm neuroscientist. I'm studying Parkinson disease, like the pathogenesis of the disease. So I have quite a lot of experience on molecular genetics. As a head of a core facility, one of the remarks I have quite often is, why are all these methods like needed? Which kind of question can you answer with these kind of methods? Like, which kind of question I have in my research could be solved using spatial transcriptomics, single cell, RNA sequencing, genome sequencing, epigenetics, and you name it. So I was thinking what could be interesting in the context of the summer school is basically going a bit toward what brings really spatial transcriptomics. How can we use it to solve some very specific question? And also, how do they complement or how do they add to other omics that we are using typically for our research? The very first thing I would like to mention, and you might already be quite aware of it, when we do research, keeping the native histological structure of our sample is quite important. One thing that one could see, for example, is in the context of studying gene expression, for example, in skin. If we do a puncture of a skin on an individual, if one would like to make single cell sequencing, the idea would be to take this puncture, trigger it in some buffers and with some enzymes, use a specific tools in order to get the sample open and the single cell dissociated. So what we can imagine from there is that this very native piece of tissue being processed for so much time in the context of enzyme, pH, and so on is not anymore exactly inside its native state. Of course, we are aware of some methods on how to limit that. We can fix the tissue. We can also like only work on nuclei that we can dissociate very fast, but all this handling has some impact on the cells and their response. Having the opportunity to take a piece of tissue, putting them like frozen in an embedding systems, like sectioning it at cold temperature and putting it on a slide in order to investigate its pattern of gene expression, it's quite appealing, like not only due to the fact that one could also look at the structure of the tissue and where are the different cells located and how could they interact, but also due to the fact that we are not denaturating the tissue at all, so working really with original histological structures. Wanted basically to illustrate my point. Why do I say that the method we are using, they are keeping the structure intact? Well, actually, there are quite a lot of different tools that we can use now for spatial transcriptomics. And if we look at them one after the other, none of them really rely on degrading the tissue, working on unlike cells, and so on. So one of the methods which is used quite a lot is the idea to have a slide of tissues and basically enrich specific transcript by capturing probes. So in this very specific case, readout would look basically that way. And having the RNA directly binding to these probes, you don't need to denature the alive structure for it. Similarly, if we would go to a fluorescence in situ hybridization using, for example, RNA probes, which are conjugated to fluorochromes, that would also enable us to look in histological structure which are not denature and detect specific gene of interest on them using this method. Some applied for in situ sequencing, where we could also amplify specific probe for sequencing on a non-denatured tissue. And same for what we called the region of interest capture. So at the end, using this spatial transcriptomic has this advantage that we keep the structure of the tissue intact, and that's basically help us not only to limit the amount of noise inside the data due to the fact that we are manipulating the samples in order to be able to analyze them. But basically, it also helps us to keep the spatial information on this sample. That means where are localized specific gene of interest in the pattern of the tissue, or could also keep us the information on how heterogeneous are some cell population in different parts of the tissue. And this is basically one of the major advantage on why spatial transcriptomic has in our hands very unique attribute when compared to other genomics methods that we have. What we are aware of and basically what is in the community like very present is the idea that spatial transcriptomics is one of the method of reference if we like to resolve tissue heterogeneity and architectures. So when this method started to scale up and started to be commercialized, we found a couple of articles in like the very big papers, which were basically showcasing, for example, in 2020, that spatial transcriptomics is going to help us to find the organization and the structure of, for example, tumor tissue, or this method could be refined with a higher resolution in order to better understand development of organism, or better understand locally how some specific cancer are either appearing or respond to treatment. But what does it mean exactly that we like to resolve the tissue heterogeneity? One of the first applications that in spatial transcriptomics is very obvious is the idea of how to basically attach to specific anatomical region of an organ some very specific and local gene expression. Here, this idea that we also called tissue atlasing is already started since a couple of years, and there are actually quite a lot of data which are already available on tissue atlasing. As I mentioned, the idea there is data bank in the context of normal tissue. What are the different structure of tissues? Which kind of different cells can we find on them? And what do the cells have specific? The beauty of this kind of method is if the conditions are well delimited, the information are very helpful, not only for a specific research project, but in general for scientists who have interest in this specific kind of tissue. So from our end, what we see now is that there are quite a lot of tissue atlas which are available on the net. I think, in my opinion, like the best one, I summarize them there. I find them the best because they are quite easy to navigate through. They also run as a web tool, so it's quite easy to question them. And also they have quite a comprehensive approach on how the data were generated and how they were analyzed. For brain, like I really like this, the brain map, basically, the Allen Atlas. This database from China, like the Stomix, is it's a very nice database. There is a lot of data sets inside it. It's not necessarily very well communicated or easily findable on Google, but it's a very valuable one. The human cell atlas is also one with more single cell, but quite a bit of spatial inside it. Protein atlas has quite a lot of experiments at the transcriptomics, but also at the proteomics level. One of the things which is always a bit of a point is, from my experience, I'm not aware that there is a very nice organized depository of spatial data. Maybe just I'm not aware of it, but I find that typically there is a lot of communication done on how to deposit genome information, how to deposit gene expression data, but for spatial, like a bit less. So the take-home message from this slide is in the context of tissue atlasing, there is already a lot of work to be done, and I think a lot of scientists could some more experiment on that already available. What is also a bit obvious, how do we use spatial transcriptomics, is the idea of snapshotting the development of a specific tissue, a specific organism, a specific pathology. There are quite a lot of methods in order to do that. The one that we encounter the most frequently is basically snapshotting a specific event at different times. So if we think, for example, about the development of an embryo, the strategy would be to take the embryo, sacrifice it at different time points, and compare these embryos longitudinally and try to interpolate or extrapolate how do specific tissue develop from specific age to another one. Here, the thing which is interesting is, to my knowledge, there are not that many methods which allow to do that in real times. For example, having a tissue culture or having cells which are being cultivated and in real time doing like a spatial transcriptomics or proteomics approach. One of the applications that quite a lot of scientists come to us and discuss with us as a core facility how to approach the problem and which kind of tools to use is connecting what we call molecular signature of pathology. So what are like specific effects that we see at the tissue level and that we can characterize at the tissue levels, and like how do they basically connect at the spatial level. So one example that I have here is an example about Alzheimer's disease. So in this very specific example, there are mice which are transgenic and which will develop Alzheimer's disease. And what we know from these specific disorders is that the region of the brain which are affected by the pathology, they are going to develop protein plaques. Surrounding these protein plaques is a lot of pathological events which are known. In this specific section that is highlighted on the right, what we can see in black are like all these plaques of protein aggregating, and which are basically associated with some phenotype at the histological level. On the left side, what you can see is a normal mouse, which don't have this kind of pathology. So what is interesting on this very specific example is what is specifically happening in this region and what are the molecular mechanisms which are surrounding this kind of events. So by defining region of interest and by bringing them on the tissue, what could look at specific transcripts which are particularly highly or lowly abundant in the cells surrounding these plaques. So this is an approach which is relatively simple. The beauty of these systems is that one can detect like histologically on the tissue region which are providing a specific phenotypes toward other region which are so-called without phenotypes. This kind of approach, we see it a lot in neurological disorders. So here I spoke about Alzheimer's disease, could be also done in other diseases like Parkinson's disease. But this is also something that can be applied to different events, for example, inflammatory disease, as we just saw in the previous talk. But it could also be done, for example, in infectious disease, for example, localizing the viral entry points, or also looking how some host cells are responding to the pathogen. In spatial transcriptomic field of research, which is slightly more complicated, but which is also growing quite drastically, is the idea that in disease, it might be that we don't have like a specific region which present a specific phenotypes, but it might be that actually inside the pathology, they are just a reorganization of cell happening. The cells are changing their function and they are changing their activity, and there's something pathological is happening. Typically, like one of the example in this direction is definitely like cancer and tumors. And the concept behind that, if we can illustrate it in a scheme, is that we have a specific tumor, and this tumor environment is a bit heterogeneous. So what we would have is a central core of the tumor, which is quite dense. We have like the tumor growing, and at the interface of the LC and tumors, there is what we call the invasive front. There are some necrotic region inside the tumor core, which are so dense that they lack nutrients and they lack oxygen in order to survive, and these core are basically dying, so the necrotic regions. And we have also like surrounding like the stroma of the tumors, which are basically all the tissue necessary for these tumors to continue surviving. When one would look at this tumor microenvironment, trying to zoom a tiny bit in, what is interesting inside this kind of environment is understanding like how do the cells communicate together, not only which kind of cell are there, but also how these cells are organized and how does these cells communicate between each other. So here is a representation, for example, of how do cancer cells are communicating together with T cells, how the exhausted T cell might communicate together with some supportive cells, and so on and so forth. So in Spatial, the idea of looking a bit more in detail, not only to pathologic events at the histological level, but also looking at the general environment of tissue on which pathology is growing is definitely something that we see increasing a lot, and applications which are quite interesting. Based on the same concept, we see also that the amount of methods which are done in order to characterize the immune microenvironment in these samples is growing a lot. And what is interesting using Spatial is that one can combine different input. One of the input would be, for example, like the classic, which kind of cell type are in my tissue of interest. But one could also combine that toward what we call a time of flight, or like cells to cells interactions. And we can combine them toward one data set where we can visualize which kind of different cells are available, how does the cells communicate, and how heterogeneous is this kind of readout. So the studying of microenvironment, we don't do it only in oncology, but this is something which is basically getting applied to more and more field of research. For example, the lymphoid organs, or also for infectious disease. In neuroscience, this is done quite a lot toward like a brain region in order to understand like how they are organized and which kind of cell types communicate together as communication cells to cells, but also in the context of neuronal circuits. And there is also more and more interest toward understanding very small part of the cells. For example, the synaptic microenvironment. So these neurons, which are complex cells, which communicate with dozens of cells in parallel, like how do their synapses work exactly? And this is something really growing up now because resolution of this method is increasing constantly. And as I mentioned, developmental biology, it's also a field where in embryogenesis, one try to understand what are like the profile of a gene expression and how do they change, not only spatially, but also through the time. So to come back a bit in our topic, I wanted to discuss a bit what brings spatial transcriptomics new and which kind of application do we get out of them. Here, one of the things I would like to highlight on my next talks is the idea is that single cell spatial transcriptomics is not necessarily a method which add only new features, but this is basically also a method which can really help us to empower data and measurements that we already do. So for example, one of the typical example that could be done on a spatial transcriptomics is the idea to resolve cell-to-cell interaction. So how cells are communicating to each other, how do we characterize them, and how can we generalize this kind of data at the level of a tissue. There are some algorithms which allow us to do that, that I would like to present very briefly. The concept behind it is quite understandable, but as usual in this model, like the difficulty is always in the detail of them. And the idea behind like the cell-to-cell communication in spatial transcriptomics resolve around building matrix. And this matrix, they are basically representing how do genes are represented in a spatial area. By having a good representation matrix of which gene are expressed in which spatial area, one could basically start to draw interaction maps between these different dots, which are representing cells in this case. And the idea there would be to simplify this equation trying to identify which cell are the sender and which are the receiver, which cells are providing a specific signal, and which cell will receive this signal and react to that. And inside this model, typically what we would search is we would search for ligands, receptors, transcription factors, targets, and bring them into a model. At the end, the model looks in a way that if we would divide our matrix, which represents our cells, we would have specific cells which are the ligands providing one. We would have the cells which are the target providing one, and we would look spatially how far apart are they. If we extrapolate this, not at the level of the algorithm, but for example, in a couple of data sets, one could apply that to different spatial transcriptomics methodologies such as SlideSec or Visiums. And what we would do out of it, as I told you, is we would break them down in spots on which we would search for our single cell in order to create this matrix of genes and our matrix of cells. Having done that, what we would look at is we are going to look at the distance between the cells, the matrix of the gene which are expressed, and we will create a first graph network of the cells which could be interacting. We will add afterward inside these networks knowledge graphics, meaning like specific genes which are known are coding for binders, which are binding for receptors, which are also coding for specific cell signaling as well as transcription factors, and put that into a model where basically we try to go from the beginning of the signal, which is our ligands, to the end of the signal, which are the transcription factors. That being brought into the models, we are going to simplify the output of that toward like a receiver senders. And typically our final matrix will look like that. We would have the receptors of the receivers, the ligands of the senders, like the interactions that we can detect into our data sets, and which will be simplified in such a ligand receptors pathway graphics. And the idea behind that would be afterwards to plot on our tissue which cells interact together and which kind of signaling do they actively use. So that looks a bit theoretical, but if you would apply it on a concrete research project, that would be relatively easy. One would have a slide, a sec result, for example, spatial transcriptomics from mouse liver, and one would try to find inside them specific pericentral or periportal hepatocytes. So this kind of classification is basically based on approaches which are relatively well established, which rely on specific gene expression for specific cell. And the idea behind that would be to have like the identification of cells which could be one or the other phenotypes. Based on that, we would look at the gene expression inside these cells. We would try to separate what are the ligands, what are the receptors, put them in a matrix of correlation and annotate on our cells what are the different pathways which are highly or lowly expressed that we can use in order to train our cell-to-cell interactions. So in this very specific example, there are like two pathways which are mainly expressed. This is metabolite of steroids, and this is the glucogenesis. One could annotate using this model the cells, like which one are the ones which are ApoB or CD36 positives, in order to visualize inside our tissue the ligand donors and the ligand receptors. At the end, when going into the signaling pathway inside it, one would realize that these are like these are like the major centers which are involved inside these signalings, and one could separate inside this tissue these very specific hepatocytes toward a specific ongoing signaling, and in this case, like the IMPK metabolism or like PPAR signaling pathway. As I mentioned, if we think about how does spatial transcriptomic can bring more into the equation and what spatial also offer that other omics might have problem to answer. Definitely, spatial transcriptomics is complementing very well other omics. So we had previously a really excellent example toward how to integrate a single cell and spatial transcriptomics. If I try to recapitulate what was mentioned before, the overall idea behind that is how to get the detail of molecular pathways that we have inside single cell sequencing and how to bring it at the spatial level. So how to see which cells are directly in contact, which cells are located in which organs, in which substructure of the tissue, or are directly connected or not connected to each other. How this is done actually is the concept is again relatively simple. The application of it is always where the complexity starts. But the idea would be to define on a specific array of spatials what are the different cell types that one could identify, and what are like the specificity of these cells in order that one could associate vaguely which cells might be where on these tissues. On the other hand, one would take like an adjacent amount of tissue, and one would do single cell out of it in order to have a very good representation of what are the transcripts which are exactly expressed on a specific cells. By regressing the two data sets from the single cell and from the spatial, one can basically get the in-depth information from the single cells toward with their location on a specific slide. So of course, this needs to be done using a statistical model. Probabilistic modeling is typically used there. And what is quite important is also to address a specific score and a specific reliance on the data which are generated in order to remap them inside the same tissue to continue doing the data processing. Application of this kind of algorithm are like really neat. One which is used quite a bit is relying basically toward spatial and single cell in tumors in order to define subtumor structures. The idea behind it is exactly the same that I mentioned. The idea of the workflow is to get the spatial and the single cell from a very similar tissue, usually two adjacent sections. What would happen is we would use the spatial in order to define roughly where are the cells located, and we would use single cell to get more information towards these cells. Well, what we would do is we will use a model in order to define where the cells are and which specificities they are, and then they would be basically re-annotated on the pictures. An example which is well illustrated here is, for example, the idea to make a cell state versus spatial ecotype map. And the output of this kind of analysis are quite nice because, for example, what we could see is that in stroma of tumors, so the area of a tumor which is like supporting the tumors, but where there is no tumor cells, one could see that there are, for example, like a very similar composition of cells, but the cells are not distributed the same in the cluster 1 or in the cluster 2. So in the cluster 1, we can, for example, see that the fibroblasts are holding the structures, but we could see that in the 2, like the structure is disrupted and we lost the kind of organization of these blue cells, which are separated from these red cells. That could be also basically applied in the context of tumors. And this is a very interesting tool and very powerful tool, for example, in order to understand immune environment of a specific tumor, testing if in specific environment there are some immune cells which might be infiltrating or which might not be infiltrating, which get activated or which get exhausted. What might be basically the cells like playing a key role into that, and what might be the signaling involved inside it. In the next minutes, what I would like to show you a bit is not anymore like application we have from spatial transcriptomics, but rather how do we complement this spatial transcriptomics information, which kind of omics are used inside it, and what are the molecular genetics tip and tricks we are using in the laboratory to solve these kind of questions. So in this specific slide, what I want you to show is that currently in spatial, there is a need to get more and more molecular information inside it. In our lab, most of the experience that we have is toward like transcriptomics and spatials. This is basically how do we use specific methods such as Visium, SlideSlick, and so on in order to visualize which genes are expressed where on the tissues. But there is an increasing amount of application which are also existing in order to detect protein, which are relying on mass cytometry or which are also relying on antibodies. There is also an increasing number of epigenomics methods which are being implemented, methods which combine, for example, attack sequencing together with RNA sequencing, spatially, or CAPTAG and RNA sequencing, which is also done spatially. And with increasing performance of MALDI off top and of Orbitraps, there are also more and more methods which are getting especially more and more precise toward measuring metabolites on tissue. So this is like the challenge in the labs which are being tackled, like how do we detect these specific molecules and how do we quantify them on the level of tissues. But as importantly, we also see that there is an increasing amount of tools which are being released for integrating this data together. For example, how could we detect spatial attack and RNA-seq on the specific sections? How can we quantify effects? How could we integrate this measurement, and how could we interpret as their changes? But there are also strategies which are coming computationally in order to transform our slides toward like 3D objects, but also how to integrate as good as possible like times inside them. As I mentioned you, most of the time these methods are done on a snapshotting. That means that we have several samples which are prepared at different time points, and there are algorithms in order to improve their integration to better understand how these dynamic processes are happening. And when we think about how to achieve multi-omics on spatial, there is definitely this classical method that I already mentioned, which is used a lot. Is basically one would take a specific biopsy, one would embed it, for example, using OCT or paraffin, and one would do cryo-sectionings of adjacent slides. So one basically have a section which are separated by a couple of micrometers, and one could integrate this section either to make different stainings to detect different kind of molecules. That could be DNA, RNA, protein, metabolomics, and so on. Or they could also use in order to make 3D structure of these specific specimens. The advantage of this method is the implementation is relatively simple, as long as all the methods they rely on the same kind of fixation and the relatively similar kind of sectioning. This method is applicable, but obviously the section which is coming at a specific part of the tissue is not exactly the same one as the one that come afterwards. So the approximations that adjacent sections are exactly the same one, need quite a bit of computing in order to be addressed correctly. Ideally, one would like to stay on the same section, on the same cells, different molecules in order to better integrate them and better understand the biology of these kind of specimens. Methods which are developed more and more frequently now are the system of what we call deterministic barcoding. So the idea would be that especially one would use specific so-called barcodes in order to visualize where are specific events happening on a specific sections. These ideas rely a lot on barcodes, and they are quite powerful methods because they allow to detect more than one specific molecule or molecule types on a section. Here is an example, for example, of RNA together with proteins, where we can use a specific antibody conjugated to barcodes in order to resolve at the same level RNA expression and protein expression. But that could be done also, as I mentioned, with the transcriptomics and the epigenomics, like with ATAC sequencing or cut and tags. So these methods are basically used in research. These are protocols that we see are being developed successfully. They are still not applied in very high throughput on commercial product as far as I'm aware. So they are a bit difficult to perform. They still rely on a lot of expertise in the laboratory to have this protocol perform and successful. Another method which is used since longer and which is a tiny bit simpler to implement is the idea of in situ hybridization. And the beauty of this system is by hybridizing different kind of molecules such as DNA, RNA, or proteins. One could basically, with a fluorescence readout, look at these different molecules on the same sample, on the same section, on the same slides. Here, the whole problematic of these systems is how to handle these fluorescence stacks in order to detect this specific molecule and how to get a higher resolution out of them. And what is currently booming is the concept of multi-omic array-based approach. So the idea is that one could use specific specific molecule of DNA which are conjugated to slides, and we can functionalize them in order to detect specific protein, RNA, DNA, and so on. So these methods, they are much simpler to commercialize and produce in high throughput. And basically, it's already demonstrated since a couple of years that these methods are relatively easy to standardize, meaning company can build product out of them and sell them for services. This is why, for example, core facility like ours are scaling up services on spatial transcriptomics, because with this approach being available, it's easier to solve similar issue for a larger broad of scientists. Yeah, exactly. And there are also other methods based on like capture of specific molecule on sections, which have the advantage that the whole piece of tissue could be used for specific staining, but which have the inconvenience that it's destructive when you need to select the region with the microscopes, and usually the resolution is relatively low. So to summarize my talk, in the context of genomics, there are an increasing amount of knowledge which is available toward generating atlas of tissue, doing histopathology of specific pathogenesis, but also understanding subcellular location of molecules and their interactions. Inside this system, spatial transcriptomics is relatively disruptive in the fact that it integrates very well with other measurements, and it gives a level of information that without would be very difficult to investigate natively these kinds of events. To finalize, I would like to take the same message than Joanna. I guess this message is relatively important. The idea is when we do basic research, we have exceptionally the opportunity to invest a lot of time and resources toward understanding very complex situations. In the context of research, we can make expensive experiments using very difficult methods that could be established and implemented by very talented scientists. So we can get a very high resolution, but it's very expensive and we need to limit it to a certain number of samples. One of the steps that we see is a bit difficult is once we spend all these efforts towards generating this data, the validation of the data using different approaches, different samples, different entity is usually a bit frustrating, because one made the findings and would like basically to report it immediately, but it's very important that we do these validations. The validations, they don't need to be done anymore with methods which are so complex and so expensive. Time to time, we can also validate them using simpler experiments. We mentioned qPCR, but we could also discuss about like, bulk transcriptomics or multiplex labelings. This is like how we should design ideally our validation. Ideally, our validation is done in a way that it could be reproduced easily in different labs who might be interested to look at the specific target we report in papers, but maybe also validate other pathway one might have neglected in the data set, which other groups might be extremely excited by. So here, I think nowadays, the challenge for us is how to formulate our results from spatial transcriptomic and single cell towards simple validation strategies, and how could we basically empower this simpler validation test in order to gain a value, for example, for large clinical cohort assessments, or for example, as we discussed, like in the clinic at evaluation of, for example, good responder or efficiency of a therapy and so on. Okay, so that was my last slide. I would like to thank you for your attention, and if you would have any question, I would be very happy to answer them.